welcome back, everybody. So we are heading into the last major chunk of presentations this afternoon. And our first speaker is Elisabeth BINDER, who's come from München, where she is the director of the Department of Translational Research at the Max Planck Institute for Psychiatry there, and doing great work building a new and great organization there in Munich. So Elisabeth, please, over to you.
Okay, thank you very much. And thanks for the invitation. So far, it's been super exciting. I think I'm learning a lot. And yeah, let's see. So I think my part here is to really talk about risk factors and how we can use risk factors to maybe learn more about the disease, but also use them for disease stratification and target development. And so we know that for psychiatric disorders, both genetic as well as environmental risk factors are very important, and that it's also important to consider their interplay. So they don't act by themselves, but in interaction, basically. So what I want to highlight here is that in the genetics area, we have made tremendous progress in psychiatric genetics. And this is a plot here that a Manhattan plot from the most recent GWAS for major depression that Brenda already mentioned. And I think this is something that, like 10 years ago, we never thought would actually happen, that we see close to 700 loci for major depression. And it actually looks like Manhattan and not like the Netherlands, like Naomi Ray used to say in the past days. So we're making tremendous progress there. But we also do know and realize that environment, especially adverse life experiences, are among the strongest predictors of risk for psychiatric disorders. And these risk factors are also complex. They can occur all through development, different durations, chronicity, and also co-occur. So we already heard about the envirome. I think that's very important, or exposome, to really look at that and to look at the interplay. And what I want to point out here is that even though we often start out in the genetics to be very diagnostic specific, they're not. So we see a lot of shared heritability in psychiatry that, again, goes for transdiagnostic approaches, and the same is true for the environmental risk factors. They're not specific for a diagnosis, as we call it right now. And so each individual patient actually has a specific composition of genetic risk and environmental exposures. Maybe this can help us with patient stratification. And so I would like to maybe give an example on how to think about the genetics of the sensitivity to adversity. So adversity is one of the major risk factors, but not everybody will actually follow and be affected by disease. And so there's been a great study from the UK Biobank where they actually looked at GWAS for major depression, but stratified whether the patients were exposed to trauma or not. And what was very interesting is that there seemed to be very different sets of SNPs that contributed to the risk for depression with and without trauma. Interestingly enough, the SNP-based heritability for depression with trauma seemed to be higher than the one without trauma, and they were actually correlated with distinct other factors. So for example, the one with trauma showed positive correlation with BMI-related traits, while this was not true for the one without trauma, while the one without trauma was correlated with bipolar disorder, and this was less true for the one with trauma. So there seem to be distinct genetic subtypes predisposing to major depression with and without exposure to severe adversity. And so, of course, this has been long ongoing, this research. We realize there are gene-environment interactions that are important to understand. And we already know, like, for example, the GWAS for major depression that I showed you, Brenda alluded to, there were 4 million individuals involved. And so when we want to go gene by environment interaction, we will need even larger N's, and we will need a very good characterization of the environment and of the outcome. And so while there were some attempts to do genome-wide gene by environment interaction studies to really get at the SNPs that confer that interaction, that has been a big challenge. And so one idea that we have been following up in my lab is actually to go a step back towards the DNA to really sort of probe gene by environment interaction on the molecular level. So adversity often elicits a response of the stress system, and one of the mediators of the stress system are glucocorticoids, and glucocorticoids are actually elements or receptors that directly bind to the DNA. So one can measure the effect of a environmentally sort of challenged response on the DNA, and maybe by this we can actually understand better the factors. And so this is just an illustration. Again, like we have stress and trauma, we have our stress hormone receptors. They are like transcription factors. They bind to the DNA and they alter gene regulation. And genetic variants are likely to moderate that binding to the DNA and maybe also all the downstream consequences. And by this, maybe also disease risk. And so there are now novel tools where we can actually probe that. And these are massively parallel reporter assays. And so what we did here, is actually we took over 7,300 sequences that have been prior showed to be quantitative trait loci for response to glucocorticoids. And we tested the short enhancer, sort of putative enhancer elements, whether they were actually altering their enhancer reactivity to glucocorticoid or not. And from the over 7,000 or 3,000 something tested sequences, we found that over 500 actually responded to glucocorticoids and that over 300 showed allele-specific effects. And so this is just, again, how we do that. So there's actually the readouts is sequencing. And this is more important. Basically, what we find here is actually that we have genetic variants that alter maybe not that much the baseline activity of the enhancer, but the induced activity with glucocorticoids. And these are the genetic variants that we're most interested in. And so what we went on is to actually annotate these variants. So we validated some of them using CRISPR. We saw that they're in very interesting regulatory regions. And I'll come to that a bit later in my talk. They're enriched in enhancers that are present across many tissues. What we also wanted to know is, since originally we selected these genetic variants not for their associations with psychiatric disorders, but because they were functional in QTL studies, we actually showed that they were associated and overrepresented in GWAS for psychiatric disorders, and actually may even be causally involved using Mendelian randomization PheWAS. We also showed that transcripts related to these variants were enriched for transcripts differentially expressed in postmortem brain across a number of different psychiatric disorders. And when we take the alleles that alter this sort of molecular stress response and sum them up to functional gene score, they influence the physiological stress response. And so this is what I'd like to spend a bit more time on. So basically what we did is we took all of these SNPs here that exclusively moderated the glucocorticoid-induced response, because in our phenome-wide Mendelian randomization approach, these were also enriched for associations with neurobehavioral trait. And so what we did, we computed a sum of these SNPs weighted by the absolute change from our molecular readout. So we had a functional gene score representative of a molecular reactivity to glucocorticoid. And we tested it in a cohort that we have currently, we have well over 400 individuals, where we have a very deep phenotyping using imaging, startle response, all kinds of different things. And so what we noted is that individuals who have a higher functional gene score, so more molecular responsivity to glucocorticoids, actually also have a higher startle amplitude and less habituation to the startle reflex. When we do a sort of psychological stress test, they actually had a higher and prolonged cortisol response. This is just for illustrative purposes here, sort of the highest quartile and the lowest quartile. And when you look at the changes of heart rate during the stress test, they actually, well, I don't think you can see that. Well, they actually also basically have the higher you score, the less you actually habituate your heart rate. And we also have data where we can correlate this functional gene score now with readouts related to the brain. So, but this is still work in progress. So it seems that we have a genetic sort of indicator of a molecular stress response that seems to translate to a general physiological difference in stress responses. And these may be things that we can actually use for interventions, preventions, and other therapeutic strategies. So this is just a brief, sort of going into the, not just trans diagnostic, but trans disease. Stress is a trans disease risk factor. We heard about that already. And so this is from the British Medical Journal, a nice overview where we can see the increases in risk for both behavioral, but also physical disorder. You can see that both childhood as well as adult in adulthood, stress exposures actually increase not only the risk for behavioral and psychiatric disorders, but also a number of other diseases like asthma, COPD, dementia, cardiovascular disease, obesity, and cancer. And so what I showed you before is that, in fact, when we look at the genetic variants that moderate this glucocorticoid response, we actually map them to enhancers. And we saw where are they actually been shown to be enhancers. They're enriched across all tissues. So it's not specific for the brain. It's there in the brain, but not specific for the brain. And so when we look at the genetic variants and which traits they're associated, so it's not just one trait. Many of these variants show significant Mendelian randomization association with many different traits. And I think this is quite interesting, and I hope you can see that slide here. So this is actually a genome-wide significant hit for schizophrenia that showed the highest significance in our Mendelian randomization approach here. And this functional, you can see that the different alleles here show different responses to glucocorticoids. One is up and one is actually in opposite responses to glucocorticoids for a histone protein here. And so this is the Mendelian randomization PheWAS output for this one SNP. And so the allele associated with less expression is associated, as we knew before, with schizophrenia, but also bipolar disorder, frequency of depressed mood. And the other one, the other allele is associated with higher educational attainment, which also makes sense. But you can see it also associated with asthma, functional lung parameters. Actually, all of these are functional lung parameters and different immune parameters. So like this one genetic variant really could impact a lot of phenotypes that we have seen related to stress, not only in the brain. So again, important to maybe know when you want to treat your patients, what are the other like diseases that they would be at risk for, and maybe prevent that. And so the hope would be that the mechanistic understanding of these risk factors can support patient stratification trans-diagnostically and also trans-disease-wise, and informed by this prevention and treatment strategy, not only in the psychiatric domain, but also include potential comorbidities. And so I would like to briefly give one example where we have such a candidate gene where there's genetic variants that actually alter the response to glucocorticoids. This is called FKBP5. And so you can see here the different alleles. There's no baseline differences, but differences in how much of that gene product is actually induced. So we see what I just showed you for the large screen. And what is interesting about this gene is that it's a co-chaperone gene that forms protein-protein interaction. And by this actually influences a number of highly relevant pathways, immune-related, but also in the endocrine system and in the brain. It's related here to different kind of pathways, including mTOR and other pathways that are relevant. So you would have like a switch that's activated by stress, but then has many downstream effects. So maybe blocking that switch that's on with stress could be of relevance. And this is a summary of a lot of research by many researchers, including us, that basically this interaction of stress and FKBP5 has been documented early on. There are genetic variants that influence antidepressant response. There seems to be a genetic and coinciding epigenetic regulation that disinhibits the gene, and increased FKBP5 in animal models has been shown to increase anxiety and decrease stress coping. And this can be blocked by a small molecule antagonist. So the question is that something that it would be worth to actually go for trials in psychiatry? So this is just one of the things that basically started the whole story, was this genetic variant where the high activation variant with more FKBP5 in the context of early trauma, but not in the absence of trauma, increases risk for a number of different psychiatric disorders. And now the question is, okay, is that interesting to develop drugs for? How do we move forward? And I think, well, this is this gap here that we often fall into. We have interesting targets, but how do we go there to actually make them reach the patients? And so this is from a paper from Tom Insel and Bruce Cuthbert. And so how do we move forward? Can we move forward? And so understanding the target. So there's some work going on understanding the isoforms and the additional regulation. The question is also, a lot of these things have been genetic. Do we see changes in the brain of patient? And the other thing is, do we have biomarkers to actually have higher FKBP5 so that we can only treat those patients with high FKBP5, as we have now repeatedly heard, this is important. And so just for some snippets of what we've looked at in a lot of postmortem brain samples from a lot of different cohorts with a number of different methods. So in fact, what we do see using bulk RNA sequencing is an increased expression in the prefrontal cortical areas in schizophrenia, but also major depression. That's also seen in different cohorts and on the protein level. And when we look at single cell RNA sequencing, we see that FKBP5 is expressed in glial cells, endothelial cells, but also in excitatory neurons. And when we look at case control differences using this single nucleus RNA sequencing data, we can see that we see case control differences for schizophrenia and major depression. These are two different cohorts that seem to be quite specific for the excitatory neurons. So at least it would be an indication that patients who have these disorders, at least a proportion of them have higher levels of FKBP5. The question is, how can we identify them? And so we have this, we have all our gene by environment interactions, we have the postmortem brain data. And what is interesting, there are specific epigenetic changes in FKBP5 that can also be measured in peripheral tissue. And several publications, and this is a very recent one from Heike To's group in Mannheim, where they related the immune cell epigenetic profile of FKBP5 to neuroimaging changes in frontal cortical areas, and actually also to stress responsivity of these limbic circuits. So maybe a biomarker of brain circuit stress sensitivity. So it would now be important to understand whether this biomarker would actually also be a biomarker for more FKBP5, or is it just a biomarker of stress sensitivity in general? And so for this, since we're looking at these enhancer regions that are not well conserved across species, we actually went on and used a humanized FKBP5 mouse that actually has been sort of generated together with colleagues at Boehringer Ingelheim. So we thought about that, and Boehringer has actually published a first paper here showing the impact of this human FKBP5 gene that includes many of the introns and human exons and none of the mouse FKBP5. And it also includes this genotype that was shown to provide these gene by environment interaction as well as the functionality. And they could show that the SNP is functional in that context and that it also alters the stress response. So we were interested in, can we use that model where you have a human locus in a mouse, but you have access to the brain and the blood at the same time, and you can actually stimulate that animal and see how does methylation change in response to the stimuli or the genotype at the same time in this tissue. How does it compare to data that we get from humans? And so this is what we showed here. So we used the humanized mouse here, and so these are the DNA methylation patterns that you can see in blood. In blue is the humanized mouse, and in grayish is the human. And so you can see in blood, it's not that similar. It sort of follows similar trajectories, but it's not that similar. But what was quite striking is that in brain, the mouse methylation pattern, the humanized mouse methylation pattern, and the pattern in the humans actually follow very similar trajectories. But what we also noted is that when we look here at the same CpG methylation level in blood and in brain, it's not that correlated. So could it really be a biomarker? I think there's more work to be done. What is interesting, though, and what we noted before, is that I showed you glucocorticoids bind to these response elements and they increase gene expression, but they also have local epigenetic effects. So they decrease DNA methylation locally. And so we've shown that in humans here, we can see there's a decrease in DNA methylation that's significant across, for example, this response element in intron 7. And so when we do the same thing that we did in humans, in mice, so we have vehicle versus dexamethasone, you can see in this lighter green that there is a decrease, a significant decrease in DNA methylation at the exact same site. So the reactivity seems to be preserved also when you introduce the human locus in the mouse. So the question is, what does this mean? It's still a bit unclear to us, but we're working on really understanding how we can use peripheral measures of FKBP5 to really give us a better indication of central FKBP5 activity, because then I think it would be interesting to start to think about drugs that target that go beyond being able to use them in animal models, what we have right now. So what I try to show you is that by using genetics and really looking at the functional implication of the genetics, and we get a better understanding of the risk factors. So it's not just it's associated with the disease, but it does something specific, how it would actually relate to increase in disease. We can actually maybe have stratification. So having a high functional glucocorticoid score would predispose you to certain reactions that could be blocked or prevented. And it also may allow us to identify new targets. And that just showed data for one, FKBP5. And I think there are promises for the target. So it's strongly induced by stress. It seems to be a hub for triggering downstream effects. There are solid animal models, transgenic animals, but also humanized models. There are first small molecule antagonists, but they're not usable in humans. There's relative consistent gene by environment data, but no GWAS data so far. It's trans-diagnostic, including medical. So we just talked about, is that an advantage or disadvantage to the current situation? There is postmortem data. And challenges are, it's expressed not only in excitatory neurons of the brain, it's expressed in a lot of different tissues. It has many interactions, partners. Do we want to block the interactions with all of them, or do we want to be more specific? Yeah, again, no genome-wide genetic evidence. Apparently that's very important for companies also to move forward. And so far, we don't have very strong biomarkers for increased expression. So this was just a short roundup about risks and targets. And with this, I'd just like to thank many of the collaborators that made that work possible. Thank you.